a method used in biochemistry as well as molecular biology to separate the DNA or RNA molecules by its size. So it is a size-based separation of DNA and RNA, which is done through gel electrophoresis. And it is achieved by moving the negatively charged nucleic acid molecules through an agarose matrix with an effect of electric field or electrophoresis. Okay, phoresis is migration, electro is electric field, gel, of course, because it is made of agarose, so agarose gel electrophoresis. Important take-home message is shorter molecules move faster and migrate farther than the longer ones. Because they are small, they can easily go through the thin paths, okay, thin pore size of the agarose gel. So this is employed basically in the estimation of the size of DNA molecules when you need to know what is the size of your particular amplicon and whether, generally when you are only checking whether this particular gene is present or not in your particular DNA sample, it is all fine. But if you are actually doing a molecular cloning and you need to have a precise size of your DNA to be inserted or to be extracted, in that case, you need to be very particular about the size of the amplicon or whatever amplification has happened. Now to check this size, Dr. Madhvi told you yesterday, we put a ladder together with our normal samples. So when the ladder keeps on running, so these ladders are actually pre-made, which are already available from different companies, New England Biolabs, for example. So when you run this, so it has smaller fragments of DNA, which will be, for example, if it is a 1 kb ladder, there will be 1,000 base pair, then there will be a 500 base pair, then 300, 200, 100 base pair of DNA segments will be there. So when you run it, they will, the 1,000 wala will be stuck at the top. Then there will be 500, then there will be 300, 400, 300, 200, 100, like this. Now, if your amplicon size is very less, you take a smaller ladder, okay? Don't take a 1 kb ladder, you take a 500 MB ladder. So 500, then 500, 400, 450, 300, 350, 200, 250, like that. So you can differentiate among 50 base pairs also. So when you have to be very precise. So like that, we can check that, okay, my amplicon is coming somewhere near the 300 size band. So my amplicon must be around 300 size. You can cross check it with your primer sequence. You remember initially you designed a primer for this particular gene segment. So when you put your primer sequence here, then you can count how many base pairs are there in between these primer sequences. So that many base pair amplicon should be there, right? So this is a cross check. Analysis of PCR products can be done through agarose gel, what we are trying to do here in molecular genetic diagnosis or DNA fingerprinting. Separation of the restricted genomic DNA prior to southern analysis or of RNA prior to northern analysis. As I told you that when you are talking about molecular cloning, it becomes very important for you to have a particular size of amplicon, which you can check through gel electrophoresis. Let us first see the principle of gel electrophoresis. Although you have, Madhvi ma'am has told you yesterday, but it will be good if I reiterate it, that there's a gel chamber, okay? And in the gel chamber, there will be a negative electrode, there will be a positive electrode, buffer chamber will be there, and this is the gel, you can see it here, okay? Then there will be 1, 2, 3 samples, okay? Now I'll just fast forward this. So around 5,000 base pairs is equivalent to 1 to 2 micromolars. This up to this size, you can actually differentiate the DNA molecules, okay? Now I've already told you about what gel electrophoresis is. Then this is a negative charge, this is a positive charge. You know DNA is a negatively charged molecule, so it will run from a negative charge to a positive charge. Check it here. When you connect these negative and positive things through a amperometer, the circuit is complete, you know, right? So when the physics again, so when the circuit is complete, the current starts running through it. It runs from negative to positive, okay? So when you are loading all your samples in this and then you run your gel, you will see that the smaller molecules will come down, then the heavier one, and then the most heavier one. just in case it is not amplified at all. For example, your genomic DNA was there and it didn't get amplified because you forgot to add the primers to it, for example. Okay, then what will happen? It will be stuck here in the gel well itself. It will not run any further because it is very heavy molecule. It's the genomic DNA, right? The complete genomic DNA. So it will not run. Otherwise, if it is smaller, then it will come here. In case of certain mutations, it keeps on running to the down, because there may be two fragments which may have generated. Okay, in case of restriction digestion, when you're doing restriction digestion, then also you can get multiple bends because your enzyme is cutting it at different places. Okay, so this forms the basis of. Now ladder, when I told you 5,000 base, 500 base pairs, 1,500 base pairs, 500 base pairs. So accordingly, you can check here the size of your amplicon. It comes somewhere between 1,500 to 500. So it is closer to 500. It may be around 600 to 700 base pairs. Okay, this one will be around 1,500 base pairs. So like that, you can actually calculate the size of your particular DNA segment. Okay. Now you see here, what is this? This is an intercalatin dye, the ethidium bromide. Okay, yesterday ma'am also told you that we use ethidium bromide to actually check how much the gel has run, and the fluorescent dye, that is the fluorescence that is coming out of it, is actually because of the ethidium bromide. What ethidium bromide does is, if you can see here in the middle, this one, this is nothing but the structure of ethidium bromide. Now these are the two strands of DNA. Okay, so it actually intercalates between the two strands of DNA. Once it intercalates, so it means that it runs along with the DNA. It is capturing all the DNA with it. So then we will know that the fluorescence, we are getting this fluorescence because of ethidium bromide. And this is the actually ladder. You can see here different bends, right? These different bands are because of the different sequences of the DNA that is coming. And this particular band, it is coming somewhere near 500 to 600, 500 to 700. So it will be around 650 or 680 in its length. Okay. Now let us see a working protocol. How do we actually work on gel electrophoresis?
Agarose gels are used in molecular biology for the separation and purification of nucleic acid fragments. A dye is added so that the fragments can be visualized under ultraviolet light. The samples are loaded into the wells of the gel and separated by size using an electric current. The size range of fragments that can be separated using agarose is between about 0.2 kilobases and 20 kilobases. Agarose gels can be made with varying concentrations of agarose, between 0.6% and 3%, depending on the size of the fragments you wish to separate or resolve. Smaller fragments are resolved better in a gel with a higher percentage of agarose, and larger fragments are resolved better in a gel with a lower percentage.
You remember Madhuri ma'am told you this thing yesterday, that how the gel concentration can actually affect the movement of your DNA molecules. Okay, so what happens is, if you are putting more of higher percentage of gel, then smaller fragments can be easily coming to that. So we generally use 1.2 to 2% of agarose gel is what we generally set for our PCR amplicon checking. Okay, and if you have to check your genomic DNA, when you are isolating your genomic DNA and whether your genomic DNA has been isolated or not, to check this also you can run it on a gel. That we will take for as 0.8%. You understood? So the lesser the percentage of gel, the more the pore size, the movement of heavier molecules of DNA will be easier. If it is a smaller size, because the concentration is high, the pore size will be small. If the pore size is small, then the smaller fragments only can move, the higher fragments cannot move. This is the only principle.
Percentage of agarose. There are two basic components to an agarose gel: the agarose, which is a white powder, and a buffer solution. These need to be heated together to make the gel.
This is how the apparatus looks like.
When preparing an agarose gel, you'll need to both weigh out the appropriate amount of agarose and pour out the appropriate volume of buffer solution, which will vary depending on the size and agarose concentration of the gel.
Okay, so one thing that is very, very important for you is to always keep your notebook ready. Okay, whenever you're doing any experiment, like this is the way I have been working all these years. It is almost around 18 years now that I'm in research. So this is the regular practice. You always keep your notebook ready with you. Before you start any experiment, plan it. Okay, just make a table, plan everything. Oh, I should have brought my notebook to show it to you, but you just plan everything with it. And what are you expecting, what is the protocol you are going to follow, should all be there on that particular sheet of paper. Okay, and preferably in your notebook, not as a loose sheet. So when you put it there, you know exactly how much you have to weigh, how much you have to mix, and what you have to do. At that point of time, you will never fail in your experiments, because you are clear in your mind that how much you have to weigh, how much you have to mix, how you have to mix. Okay, write every minute details. For example, when you are pouring the tea, and if you poured a 2 ml extra, write it there. No harm in it, just write it there. Because if you fail in the experiment or you get very good results, you know what changes you have made in it. I generally used to write my protocol in black pen, and then wherever I'm making some changes, that particular step I used to mark in red pen. So I know that what changes I've made, and then whatever result you get, paste a picture there, and then you write what is the inference. Okay, even if you fail, paste the fail picture. You don't get a gel picture, just put a blank gel doc image, and then you write it there. No bands were seen. So it will be very good for you as a good practice.
Combine the agarose and buffer solution and mix them to make sure that no large lumps of agarose powder are stuck to the container. The next step is to heat the agarose and buffer mixture in a microwave oven until the agarose is completely dissolved. Make sure the container holding the gel mixture is large enough to allow for the solution to boil up without coming out of the container. A loose cover should be placed over the top to prevent the solution from splashing out. It's very important that this cover be loose.
You heard that? It is very important for the cover to be loose. You remember, you must have done this autoclaving as well, right? In microbiology or cell biology media or something. What we do is, we don't tighten it properly. Why? Because the heat that will be generated, the vapors that are coming out, chemistry. I told you it's all multidisciplinary. Okay, so when the vapors come out, it should not blast the substance.
Otherwise, dangerous pressure buildup can occur within the container. You should mix the gel solution at intervals during heating, but be sure you wear safety gloves. The solution can become superheated, and when you mix it, the liquid can suddenly boil up. The agarose is only fully dissolved when it's completely clear. If you don't completely dissolve the agarose, your finished gel will have regions of different concentrations, and your nucleic acid samples will not separate.
Very important step. This is very important. We really feel that it is nothing, but we just have to mix and we have to just heat it or boil it. I generally do it in a conical flask. Okay, and when you are mixing it, you have to be very proper, very thorough, and like it showed, I'll just show you again. You see here, if you see like this, heat it a little bit more or mix it a little bit more. Okay, but very careful, don't try to mix it with your bare hand or with only little latex gloves. Don't do like that. Okay, wear silicone gloves or wear the microwave safe gloves.
Fully dissolved when it's completely clear will have regions of. Once the agarose is fully dissolved, let it cool to 60 degrees Celsius. This prevents heat damage to the gel tray while keeping the agarose liquid.
Now you may ask that how do I know when it has reached to 60 degrees? It comes with experience. Frankly speaking, no rule or no charge to it. So it comes with experience. So when you are initially, when I used to do, many of the times I actually destroyed 2 of the gel trays. So I'm very ashamed of it. But this is bound to happen. So if you worry that you may fail or you may do any mistake, you can never succeed. So just follow the instructions, but at the same time, don't worry if you are not, completing it to the full. So generally the setting temperature for agarose is 45 degrees generally. There are two types of agarose. When one is low melting agarose and one is the regular agarose. The regular agarose is what we actually use here. We don't use the low melting one. Low melting is used in making devices, which I will be telling you if time permits. I'll be telling you about how we are making the devices roughly orally. So just keep it and just see if you can feel the touch of it. Okay.
The agarose is cooled, you'll need to add a dye to the molten gel for later visualization of the size-separated nucleic acid fragments. You can use cyber green, but the best and most commonly used dye is ethidium bromide. When using ethidium bromide, you should always wear gloves, since it's a mutagen and a possible carcinogen that can...
Okay, so when you are handling ETBR, you should never handle ETBR with your bare hands. And not just that, once you have added it to the gel, you should never touch the gel also with your bare hands because it contains that ETBR. Okay, be very, very careful about it. It is a carcinogenic material. I told you it's an intercalating dye. So it can intercalate your own DNA in the cells. So that ways you have to be very careful. You don't mishandle certain things or chemicals.
Be both absorbed through the skin and breathed in. Immediately after handling ethidium bromide, change your gloves to avoid contaminating other instruments or parts of the lab. Gel trays have two open ends that need to be sealed while the gel is solidifying. This is easily achieved using masking tape. Take a piece of masking tape that's a few centimeters longer than the open end of the gel tray and fold over one end of the tape to make it easier to remove later. Place the tape around the end of the gel tray, overlapping both sides of the tray, and make sure it's firmly stuck along the entire end.
Don't feel that they show something in the video and when we are doing it is different. It's not like that. I do exactly the same thing. The only difference is I don't use the masking tape. I'm using the regular sellotape. Okay, the regular sellotape that we will get in our laboratories or in the house. The regular transparent tape also you can put. I generally put that.
Do the same for the remaining open end. Before pouring your gel, you need to make sure the gel tray is on a horizontal surface so the gel will be of uniform thickness. You also need to insert a comb into the gel tray. The comb will create cavities called wells. It's into these wells that you'll be pipetting your nucleic acid samples. Once you've made sure that the gel tray is level and you've inserted the comb, slowly pour the gel solution into the gel tray.
Did you listen to it carefully? Slowly pour, because we want to avoid any bubble formation. If bubble formation is there, what will happen? It will interfere with the movement of the DNA along with the gel.
Using a disposable pipette tip, move any air bubbles to the edge of the gel. It's especially important to remove bubbles from around the combs since these can affect the shape of the wells. It takes about 20 minutes for the gel to solidify, during which it will change from colourless to slightly opaque. While the gel is setting, don't move the gel tray or disturb the gel itself since this can create a gel of non-uniform thickness. Once the gel is set, you can remove the comb. Slowly and carefully pull the comb upwards vertically. You'll feel some resistance as the comb comes out of the gel.
This is a crucial step. When you are removing the comb, be patient. Slowly remove the comb. You may have to slightly move it from the sides and then remove it, because if your wells are actually damaged, then the movement of your DNA will be lost. You will not get a clear sharp bend. You will get a very haphazard kind of bands, U-shaped, V-shaped, sometimes W-shaped. So all this happens.
Then remove the masking tape from the ends of the gel tray.
And now your gel is actually set. Now you can only, you remove your gel. Don't leave your gel tray together with the masking tape in the buffer solution, because it will never run. because it is, it is having.
Agarose gel. Agarose gel.
So now we will see how we are loading the samples into this agarose gel. See, now this step we have already seen. I told you, right, I was doing 96 samples in one particular cycle. So if I'm doing 96 samples, I have to do checking also for the gel checking also for all the 96 samples. So for that checking, now here you can see there are two combs, right? Two white combs there, because the size, maybe I just need to check whether it is amplified or not. So it doesn't matter if they are resolved more or not. So I just need this much of space for one particular sample. So I can use the same tray for two different more number of samples in one go itself. So once you have poured it, then you have set it. Now you have removed it. Add your sample into the gel one by one, and you, before adding into the gel, you have to actually load it with the loading die. Okay, loading die, because you need to know how much it has run. The loading front, the running front, you need to know. So for that, we put it in the loading die, and then we add the running buffer. Running buffer, because electricity is to be conducted through that, right? So we have to use the buffer. Now we are actually adding the ladder in the first one. Okay, because we need to check the size in both the combs. Now we are adding the samples. Now you see how it is being added. You just take the tip till the end of your well. See this. We don't know that this is a well itself, but when you are putting your pipette inside, it will go all inside that well. Just see. Very carefully you have to put. In fact, agarose gel is much more easier to handle than the polyacrylamide gel. Okay, so if you are using this particular gel, regular PCR and checkup, it's fine. But for page, it will be a little difficult or tricky to actually load your samples. Yesterday, Dr. Madhuri told you about that, right? So this is how you actually put your samples inside. It may sometimes it may come out like this also, but don't worry about it. You may be losing a little sample, so it is better to be careful rather than because when you lose the sample, you may lose the fluorescence as well. So when you are checking, although the expression level will be the same, the amplicon will be the same, but the sharpness of the band will vary. So this generally we do when we are checking the effect of any compound, as I told you. So concentration dependently, if the compound is having some changes on the DNA, that we see through the fluorescence only, through the amount of DNA that we are actually amplifying. But if because of your mishandling, it did not come out, you will be having a wrong result or a false result. Okay, so this is how you add the samples. Now coming to the next step. Once you have made your gel, you have put your sample, you have run your sample, and you get the bands. Okay, now you need this particular DNA for sequencing. Most of the times what happens is, I told you that we have put 25 microliters initially in the PCR reaction. Just a second. I told you that we have put 25 microliters as the reaction mixture initially. Out of that, only 10 microliters we have used to put it for the gel checking. Okay, for example, remaining 15 microliters is still there. So with that 15 microliters, we can put a sequencing reaction. It's completely fine. Just in case you have put only 10 microliters in your PCR reaction, and then for all the 10 microliters you have added, or you want to purify, this is one way of purification of DNA as well. So if you have put it in the gel, then you need to recover that particular DNA, right? Because you will be losing it. How will you do a sequencing then thereafter? So for that, we need to do a gel cutting or the gel extraction, DNA extraction from the gel. So we do it on a transilluminator. You look at it here, this is a transilluminator, and you always wear your gloves because the gel is actually nothing but ethidium bromide incorporated. So don't use it without this thing or without a gloves. Okay. Now you take a scalpel, take this gel, put it on the transilluminator, and switch the light on. Light in the sense that you know that there will be two wavelengths in a transilluminator. So when you put it on a transilluminator wavelength, you can see the bands here. Look at this. Look at these. Can you see? And with these bands, we can start cutting with the scalpel. We cut it here. Slowly, you don't want to, give any problem to your transilluminator as well. And then you see here, these are the two bands. You can see fluorescent bands. These are the two things that we have actually cut it from this particular gel. Now this we will be collecting, and remaining thing we will be throwing away, and this we will be collecting and cleaning our transilluminator. See here, another gel. If you can see 1, 2, and so this is the ladder, and these are our two bands. Now I don't want this smaller band, I want this bigger band. Okay, so slowly we will be cutting this bigger band. You see here. Remove whatever that you don't want and just take only the bands. Can you see this? Beautiful. This is very beautiful. See, the complete DNA that was there in that particular amplicon is here now. So now we take it and we elute it in the, we melt the agarose and elute this DNA in our TE buffer. Okay. Now, we have this DNA in our hand. We have the DNA PCR amplicon in our hand. What we will be doing now? We will be doing a sequencing. We need to know what is the sequence of this particular DNA that we have actually extracted and we have actually amplified. Whether whatever we amplified is there or not, we need to sequence it. Okay. And now, if for example, when I talk about genetic mutations, so this is a particular gene sequence. Okay. This sequence has to be there in the particular segment. But a mutation here, or a single nucleotide point mutation, SNP, if it is there, then this thing will not be there in my sequenced amplicon, right? In case if I am a diseased person and you are sequencing my DNA, my sequence and the parent sequence may not match now, because there will be one mutation here. That's how I'm different. So to check these mutations, we do generally sequencing. So what is sequencing? DNA sequencing is nothing but a process of determining or identifying the order of nucleotides present in a DNA sequence. Okay. You know that the concept of triple codons and triplets, and then the amino acids. Triplet codon actually codes for an amino acid. Suppose there is one point mutation, then the triplet codon may be changed. There may be a shift mutation or something like that. So all these things can be checked through DNA sequencing. So for understanding the structure and function of a gene, it is very important to study the nucleotide sequence of it. And the DNA sequencing serves this purpose in a sequential manner. The long chain of DNA is actually read by the sequencer. It is just like you are walking on the road and checking or counting the bricks. It is just like that. The sequencer or the probe molecule actually walks around the sequence and then keep on adding one and one sequence and then telling us what is happening, what is the sequence of this. So the amplification process in the sequencing is similar to PCR. However, here the nucleotides are either radio or fluorescently labeled. Okay, that is the difference. Apart from the regular nucleotides, these nucleotides are actually DDNTPs. Okay, dideoxynucleotides. Generally, we are using DNTPs in the PCR. That is the deoxynucleotides, right? In this, we are using dideoxynucleotides. Here, what happens is on the third place at the sugar level, the OH is actually converted into H. So it is dideoxy. There are two deoxy molecules. So when generally in normal nucleotides, what happens is this OH actually sequesters another nucleotide to add. Okay, keep on adding and making a chain. When it is dideoxy, what happens here is it doesn't have a hook. It cannot hold on another nucleotide. So if it cannot, here the chain stops. So all the 4 nucleotides are actually differently labeled. Different colors will be there to it. So as soon as it keeps on stopping, we know that this sequence has stopped here. This sequence has stopped here. So all these collectively form the sequence of that particular DNA, and we check for it. Now, there are many methods of DNA sequencing, but the most commonly used is the Sanger's method of DNA sequencing. Now, let us see what this method is all about. And I'm again using the audio of this particular video because it is very self-explanatory, and I'm sure you all will understand better in this. If you have any queries, you can ask me later.
What do we mean by DNA sequencing? It is a technique by which the precise order of nucleotides in a DNA segment can be determined. Today we are going to discuss original Sanger method of DNA sequencing. This method forms the basis of modern computer-automated sequencing techniques. Sanger sequencing was developed by Frederick Sanger and colleagues at the University of Cambridge in 1977. This technique involves in vitro DNA synthesis. It is based on the principle and biochemistry of DNA replication.
I told you the same thing about the PCR, okay? And as I told you initially in my previous slide also, sequencing works on the similar principles of the PCR.
Tell me, what will be the basic requirements for in vitro DNA synthesis? First, we should have a DNA template strand. This strand will provide complementary bases for the synthesis of the new strand. Here, this is 3' end of template strand and this is 5' end. Next, we require a primer. As you know, a primer is a short oligonucleotide sequence, complementary to the sequence, at the 3' end of the template strand. It serves as the starting point for new strand synthesis. We will also require deoxynucleotides. To catalyze the DNA synthesis reaction we require DNA polymerase. DNA polymerase incorporates deoxynucleotides complementary to the template strand and extends the DNA chain in 5' to 3' direction. Now here we need to note very important point. Primer is one of the essential requirements of DNA synthesis. But why? Because DNA polymerase cannot catalyze the synthesis reaction on its own. It requires 3' hydroxyl group to form phosphodiester bond with the incoming deoxynucleotide. And this initial 3' hydroxyl group is provided by the primer. Once DNA synthesis is initiated, each deoxynucleotide incorporated in the growing DNA chain has a hydroxyl group at the 3' position of the deoxyribose molecule. And thus, DNA polymerase keeps on elongating the chain. Sanger DNA sequencing technique makes use of modified deoxynucleotides known as D-deoxynucleotides. Now, what are these deoxynucleotides and what is their function in DNA sequencing? Look at this image. This is a basic chemical structure of a deoxynucleotide. Here, you can see that, at the 3' position of sugar, hydroxyl group is present. This is the 3' hydroxyl group which participates in phosphodiester bond formation during DNA synthesis. Now, look at this image. Here, the hydroxyl group is absent at the 3' position of the sugar. Instead, there is a 3' hydrogen. This is deoxynucleotide. If in a DNA synthesis reaction, a deoxynucleotide is added, the DNA synthesis will terminate with the incorporation of this deoxynucleotide. This is because, now there is no 3' hydroxyl group for the further extension of DNA chain. For this reason, deoxynucleotides are called chain termination nucleotides. And, Sanger technique is also known as chain termination method or D-deoxy-DNA sequencing. So, if the concept of D-deoxynucleotide is clear to you, it will be easy to understand the procedure and interpret the results of Sanger sequencing technique. Let's now move on to the Sanger sequencing method. Sanger sequencing consists of 4 separate reactions that run parallelly. We will label them as 1, 2, 3 and 4. Each of these reactions has some components that are common to all. These common components are: First component is DNA template strand. Many copies of this strand are used in each reaction. Let's say this is our template strand. Second component is DNA primer. Again, many copies of DNA primer are used. Sanger Radio labeled these primers for detection purpose. Third component is DNA polymerase. And fourth component consists of all 4 standard deoxynucleotides or DNTPs. They are used in large amounts. Besides these common components, small amounts of 1-deoxynucleotide or DDNTP is added in each reaction. This is the component which is different in each reaction. So, for our illustration, let's say DDATP is added in the first reaction, DDCTP in second, DDGTP in third and DDTTP in fourth. Now, as we know, D-deoxynucleotides or DDNTPs, when incorporated in an elongating DNA chain, they will terminate DNA synthesis reaction. Since small amounts of DDNTP are included in each reaction, DNA synthesis will not terminate every time. But DNA polymerase will occasionally insert a DDNTP, instead of a DNTP, into a growing DNA strand. Since the incorporated DDNTP lacks 3' hydroxyl group, it cannot form a bond with another nucleotide. And DNA synthesis will terminate. Thus, in each case, we will get partially replicated fragments. Let's understand this for the first reaction. This is our DNA template strand and this is the primer. DDATP is present in lower amount. DDATP will be incorporated occasionally wherever normal DATP is to be incorporated complementary to thymine residue. And each time a DDATP is incorporated, the DNA synthesis terminates. So, as you can see, there are 3 thymine residues in the template strand. Complementary to these thymine residues, DDATP can be incorporated in the new strand. At the end of this reaction, there will be 3 partially replicated DNA fragments. Similarly, 3 partial replication products will be produced in second reaction, where DDCTP will be incorporated occasionally, complementary to guanine residue. In the third reaction, 2 partial replication products are produced. And in the fourth reaction, 4 partial replication products are produced. Upon the completion of
So I hope you have understood what this Sanger sequencing is all about. So let us now see how do we actually prepare our samples for sequencing, and then how we are going to do the sequencing. See, I told you that when you are getting your PCR product, you need to actually clean your product or to prepare your product for purify your product for sequencing, because many of the things in your PCR mixture may interfere with your sequencing reaction. Okay, so when you have your amplified sequences, many of the companies are actually also giving some spin columns. Okay, so this is another chemical, that is exosap, or there are PCR pure. There will be many chemicals like that which will be available from the company. So we actually add it to our mixture and centrifuge and remove it later. You see, you are adding it here in your samples. Just check this. Okay, so just like the initial phenol chloroform extraction method of DNA, it will be taking off all the things, leaving your DNA. The same thing will happen here. This all cocktail will also remove all of the chemicals that are available in your samples, and once you centrifuge it and you take out all your substance from there, incubate it for 16 minutes so that it can actually chelate all of the compounds, and then you will be left with your DNA samples in it. Okay, so now we are going to, as I told you initially, we will be centrifuging it. After mixing it, vortexing it, mixing everything together, just put all of your samples into the centrifugation. See, we are preparing our samples for our sequencing. So we are taking all the reverse sequencing primers, forward primers, everything we are keeping together with us because we have to next go for the sequencing of it.
Dear madam.
Yes, ma'am.
If at all you want, you can take 5 minutes break, madam.
It's fine. It's good that we are going in a flow. So I just thought that we'll finish the sequencing and then maybe we can take a break before molecular cloning.
Yeah.
The sequencing, then you can go for the break.
Yeah. So we are adding the molecular grade water, and we are doing it for all of the samples that we have added, and we have purified them. Now our samples are ready together with the water for sequencing. Mix everything very thoroughly. Now here you can use the same tip because now you are not going to do separately, na? So you can use the same tip for different triplicates. Now when you have your reverse primer and forward primer together with the water, we have to mix all of these things together to be put in the for our sequencing. Okay, so now in this we are putting our primers, forward primer, and then the reverse primer. Mix in all the samples thoroughly. And once you have mixed it, we keep it aside for our sequencing. Now, how do we actually do the sequencing? I'll just show you a video for that. This is all how the sequencer actually looks. Okay, so all these things you have to actually change in the sequencer. So what are ABI? ABI is the company which is generally providing all the sequencers to most of the labs. So we are using these ABI sequencers in our labs. So most of the times all the programs will be similar for it. For us, we have to change all the programs depending upon our amplicon size, depending upon our sequence. All these settings can be done once you have, you know what you are going to actually amplify. So just see here, you can see the sequence, the number of sequences that will be available. I told you, right, for the different DNTPs, there will be different sequences that will be generated. So with this, we can have the different sets of sequences and we can compile them together and you get this kind of sequence. See this, you can see different colors, blue, green, red, as well as black. So these are the 4 colors that the DNTPs are actually tagged with. So depending upon whenever you are getting something, you keep on getting the sequence. Now, the sequence may be around 2000 base pairs long. Always the sequencing primers are designed a little ahead of whatever your primers were. So when you are getting this particular sequence and you need only this much, you need to crop the sequence. Okay, this is all the analysis from the ABI software. So if you have the ABI software in your college or in your research institute where you are going for further research, you can do it this way. Okay, I don't think that until unless you do this particular analysis, you will not be able to get it properly. Howsoever we give you a demonstration about this technique, but until unless you sit on your max for this, you will not be able to do. I have done it for 3.5 years, and it is tedious, but it is every single, you can see here, every single base pair you have to actually read. So it becomes difficult when your amplicon size is a little bigger. Okay, so this is how you analyze your sequence data. Okay, so the last thing in sequencing, I want to show you how I have used this sequencing in my particular work. So this is a work way back, 11 years back, I published this paper in 2009. So this is the latitude-wise prevalence of the CCR5 delta 32 HIV resistance allele in India. So this was a population genetics study that I did. There is a 32 base pair deletion in a particular chemokine receptor in our body. So this receptor is actually used by the HIV virus to enter into the cells. Like how you know that ACE2 receptor is used by coronavirus to enter into the cells. CCR5 is the receptor, chemokine receptor 5 is the receptor which is used by HIV to enter into the cell. Now, if there is any alteration in the sequence of this particular CCR5, the virus cannot enter, right? So I need to check that whether all the individuals are having the same makeup of CCR5 or there is some deletion mutation, because this particular 32 base pair deletion is known to confer resistance to HIV infection. Okay, so this is the work that I did, and you see here, this is, I have marked it in red here. This is the 32 base pair that were deleted, and I did a sequence analysis. You can see down, you can see this page. The Agarose Gel Electrophoresis, where the wild type or the single mutant, there is no mutation, it gives a single base pair. And when you see the heterozygous, you see there are two bands there, right? And both the bands, you can see here the difference, it will be more or less than 32 base pairs only. So this is the exact sequence that is not available in some particular individuals. And you see here, this is the clean sequence in the normal human, and this is the sequence with bland lines or flat lines, because there is no sequence to be read there. So this is the value of sequencer or the PCR that we can actually